Okay, so you should have plenty of time in this lab to go through the homework with the students. So I want to make sure you're familiar with it, as always. So the first part of the homework is talking about seeds and seed germination. Okay, so let's say we take a seed and we plant it in the ground. And again, they're going to be working on this individually, and you're going to go around and help the groups. You're not going to have like a class discussion on the homework. You're going to be helping them individually. But we're going to take a seed and we're going to plant it in the ground. So there's our little seed. And the little baby plant is inside of that seed. Okay? And so the first question asks, when it's in the seed and it's down in the ground, or it's not even planted yet, what metabolic processes are happening? So it's inside the seed coat and the seed coat is closed. So if the seed coat is closed, they're not getting any oxygen and they're not getting any carbon dioxide. They're stuck inside the seed coat. And so they may not know this yet because we haven't covered cellular respiration yet, but there are minimal processes that can happen when there isn't any air. So think about you when you hold your breath. There are processes you can do to get a little bit of ATP to keep you alive. And so when it's like that, it is only doing fermentation. Okay, that's that minimal process. And they don't need to know what fermentation is exactly, but they need to know that it's a minimal process. Like I said, we're going to learn this in lecture right after this, so they'll be able to go back and, I understand this, and fill it in before it's due the next time. So fermentation is the process it's doing there. Once you add water to the plant, so once water penetrates the ground and penetrates the plant, it's actually going to break that seed coat open. The seed coat will swell and it will crack open. So once the seed coat cracks open, then the plant has access to air, to oxygen. But notice it's deep underground, so it doesn't have access to the sunlight. So what it's going to start doing is cellular respiration. And it will no longer be fermenting. There's no reason to ferment. And so in the second one, it's only doing cellular respiration. And that, what it's going to do is it's going to use the little bit of sugar that's in the seed. So there's a little bit of sugar in there. It's going to use that little bit of sugar to do cellular respiration and to grow. So it's going to grow its little roots down. It's going to grow its little cotyledon up. And as soon as it penetrates the surface, now we have sunlight. So once sunlight hits it, then it can grow on its own, right? And so once sunlight hits it, it's now doing photosynthesis. Is it still doing cellular respiration? This is a good question to ask students. The answer is yes. It never stops doing cellular respiration. It does cellular respiration just like we do. It just makes its food first with photosynthesis, and then it eats it. We just eat it, right? So it's doing photosynthesis and cell respiration. Okay? It's doing both of them once it reaches the surface. So you might pose the question to students, what happens if I plant my seed a little bit deeper? So it grows its little plant, but it runs out of sugar. So if it doesn't reach the surface before it runs out of sugar, it'll die. So sometimes when you plant seeds, you're like, what happened? It never germinated. If you dig a little bit down to the dirt, you'll see these little plants that were struggling to get out, but they just couldn't because they ran out of sugar in the seed coat, inside the seed. All right, so that's the first couple of questions, 4 questions. The next one is their cellular respiration exploration, okay? So it says for them to do a demonstration. If it's available, you can light something on fire and then put a jar over top of it. I'm happy if you do that. If you want to go grab a jar, light something, set it on something, so it's not on the table, and then cover it, and you'll notice that it goes out. So it says, what's the paper made of? Tell them to Google it. What's paper made out of? Well, it's mostly cellulose, okay? So we look at paper. Oops, that's on the right side. Paper is really big, long carbon chains. And it's actually glucose monomers hooked together, right? Oh, I cannot draw a glucose. Anyway, so that's what it looks like. Okay, that's cellulose. Number 6 asks what's happening when it burns. They should know this from calorimetry. So they should know that when you're burning something, you're actually separating bonds between our atoms. Okay? And then it says the chemical process you just witnessed is called combustion. And here we have our formula for combustion. So we're taking our carbon molecule, which is CXHY, because we don't know how long it is, and we're adding oxygen to it, and that's coming from the air, and then out is coming the exhaust, which is CO2, and water, who knew, and heat. I need to make sure you can see this on the board. Am I too high? Oh, I'm good. Okay. All right, so that's combustion. It asks them, is this process endergonic or exergonic? So that's number 7. Well, we're getting heat out, so it's an exergonic reaction, so they should be choosing that diagram. It looks like that, all right? And the energy of activation, you might point out to them, is I lit a match to get it started. Okay, so now the process, it turns out that the process of cellular respiration is the same as combustion. That's why we call it very similar, right? So it's cellular respiration is just like combustion. But it's more like a fluorescent star, where we're bringing that energy down really slowly rather than all at once, okay? And the reason is we don't want to explode, right? So we're going to bring it down slowly, but it is this same process. And so, let me see, what do we want to say here? So in the first one, it's giving them some rules. So our equation, I'm going to erase this, our equation for cellular respiration is very much the same as this, except instead of a CXHY, we have glucose. Okay, so that's glucose. We're adding oxygen that we breathe in, and we're getting out CO2, which we breathe out, and we're getting water, and we do get body heat out of it, but mostly what we want is ATP. We want storable energy. And they haven't, let's see, they're going to learn about ATP actually in lecture. Okay, so they'll learn about ATP before this. So that's what we want to get out of it. So then it's got a bunch of rules. So rule number 1, in order to not burst into flames, we want to only break one bond at a time. Okay, so that's rule number 1. And so for rule number 1, I've given you a demonstration. So we've taken a 6-carbon sugar, which is glucose. Okay, that's glucose. And we are breaking a bond. Now remember, that bond contained 2 electrons. That's what was there in that bond, in that covalent bond. They were sharing 2 electrons. So this is an imperfect analogy, but it works really well for this. So we're actually saying that once we break that bond, what comes out of that bond is 2 electrons. Okay, 2 electrons are coming out of that. Now it's obviously a rearrangement with other enzymes and things like that, but ultimately when you break that bond, you're getting out 2 electrons. And then I ask students, what kind of electrons are those? Are those electrons that are held tightly to a nucleus, or are those high energy electrons? Well, it turns out those are high energy electrons, jazz hands electrons, okay? And those electrons are way out here. Now, you might ask students, how did they get way out there? How did we get high energy electrons? Well, if you've watched the photosynthesis video, you know how we got high energy electrons. The plants put them there during photosynthesis, using the sunlight. So we are simply taking the sun's energy out of that bond, and we're bringing it back down. Okay, so these are high energy electrons. Now, if you let a high energy electron go, it's going to go like this. We don't want that to happen, because number 1, kaboom, explosive release of energy, and number 2, we just wasted that energy. So we want to take those high energy electrons and we want to store them temporarily in another bond so that we can use them later. Okay, so we have a molecule that's for temporary storage of electrons. It's called nicotinamide adenine dinucleotide, or NAD. And NAD has an available spot for an electron. It has a positive charge. So we take that and this hydrogen ion that also is a plus, so it has an available spot for an electron, and we take the 2 electrons that came out of this bond, so out come 2 electrons, and we simply put them back into a bond between NAD and H. So this becomes an NADH molecule. Okay? And think of NADH as sort of like a purse that you're carrying your little electrons in until you're going to use them later. Okay? So that's what's being demonstrated there in box number 1. I'm going to make sure this is still taping. Okay. So, rule number 2. It says these high energy electrons that have been, they have to be stored, I just went through this, they have to be stored somehow. So they're going onto this NADH, all right? So that's what you're going to show them there. So then number 8 asks them, go ahead and finish breaking all the bonds. So they're going to illustrate breaking the rest of these bonds. So you can see I got to break this one, I got to break this one, I got to break this one, I got to break this one. And every time I break one, 2 electrons come out. Right? And every time these high energy electrons come out, we don't want to lose them, we don't want to explode, so we're going to simply store them in another bond. Okay? between NAD and H. NAD and H. NAD and H. NAD and H. Right? So at the end of box 2, they should end up with 1, 2, 3, 4, plus this one, so 5 NADHs, so 10 stored electrons, and 6 individual carbon molecules. So that's what it asks them in number 9. They're going to have 6 individual carbon molecules, each of these, and they're going to have 5 of these NADHs, or 10 stored electrons. All right? So now that we've broken all the bonds, nothing else is left except for these high energy electrons, the jazz hands electrons that are stuck in the bonds right here. So what do we do with them? Well, we could pull them out of the bonds and they'll fall really quickly and will explode into a burst of flames. Or we could take those electrons and bring them down one step at a time like the fluorescent star. So something that's fluorescent, and I don't know, it depends on whose lecture you're in, but let me draw for you really quickly what happens with a fluorescent star. Right? So here's our atom, their fluorescent atom, and we've taken electrons that would be here, and we've used the light, you charge them up, we use the light to pop them out like that, and then when you turn the lights out, these are cool atoms in that they don't let it come down immediately. They let it come down very slowly, one step at a time. And when it comes down, it gives off this little burst of light, which causes it to glow. Okay, so that's different than combustion, where we bring it down right to oxygen all at once and you get that burst of flame. That's what's happening when combustion occurs. We're taking these high energy electrons out of the carbon bond and we're taking them immediately to ground state on an oxygen atom, and that's causing the big burst of energy that you see as flame. So we don't want to do this because we don't want to explode. So we want to do something, a process similar to that. Okay? So if you read rule number 3, it says that these stored electrons are very high in energy and quite explosive. So we're going to take them down one step at a time. And I've sort of illustrated it there, something like this, where we're bringing it down one step at a time. And so one way to accomplish it, okay, more hints, the energy that's extracted each time, instead of glowing in the dark, although that would be really cool if we glowed in the dark, we don't want to glow in the dark. So each time one of these falls, instead of glowing in the dark, we're going to use it to do work. So it says we're going to use it to shuttle a hydrogen ion across a membrane. So we're going to do active transport. And they should have learned about active transport already in lecture, that active transport requires ATP, some kind of energy. So we're going to actually use the energy from the falling electron to shuttle these hydrogen ions across. Okay, so let's draw that. So we've got our special membrane here, and we have little hydrogen ions. This is a hydrogen ion, okay? So we have these hydrogen ions, and we're going to use this explosion of energy, okay, that comes out when the electron falls to shove that hydrogen ion across. So each time the electron falls, we're shoving the hydrogen ions across the membrane. Okay, and given enough of these glucose molecules going through the process, you're going to end up with a situation like this, where you have a ton of hydrogen ions on one side of the membrane, right? And so that third point says if hydrogen ions flow back down their concentration gradient, they go through a special channel that can make ATP. So as we push them across using our electrons, push them across, then we have our special protein right here that'll allow them to go back through. So as they go back through, this guy is going to crank out ATP. And we'll talk about how in class, but that's the general idea, that we're using this chemical potential, okay, this electrochemical potential, to drive this functioning of this protein. It's like paying the toll for the protein to make ATP. And then the last point is the final electron acceptor. So as it's falling down, boom, 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 it then jumps onto an oxygen. And we'll talk about where and how that happens inside of the cell in class. But I want them to get the general gist of the idea, okay? So they can draw their proposal below. And then the last few questions say, which box above represents glycolysis? They may not know the answers to these. In fact, they will not know the answers to these. So don't have this, the students can try them, but they're going to go to class and they're going to learn, and then they're going to go back and answer these. So it asks which box above represents glycolysis. That's actually box number 1, the first break of the glucose, right? Which box represents the citric acid cycle? That's the remaining breaks. That's box number 2. Any missing steps between? Well, that's the addition of acetyl-CoA, another carbon gets broken off, moving inside the mitochondria. There's a bunch of steps. They can list whatever they want. But like I said, they aren't going to know the answers to 11 to 15 until they go to class. So if they don't have 11 and 15 filled out, totally fine. But they need to be down to this proposed way of doing things before they go to lecture. Okay? And then 14, which box represents electron transport chain? That's this. It's going to be box number 3. And are there any other differences? They can list a million. That it's not really doing what it's doing here. It's lots more complex. It's not falling on the same atom. It's falling on different atoms in the electron transport chain. There's a bunch of differences. But I want them to get through number 10 before they come to lecture so that I can just jump right in from this to teaching them cellular respiration. And speaking of, I'm also going to send you a video of me drawing cellular respiration. Let me emphasize again, you're not going to draw that in lab. That's way too much information. We will do that in lecture, but I want you to be the expert in cellular respiration. So if they do have questions, or if you've got a smarty pants student that wants to know a little more detail, then you'll know the detail. Okay? And I think that is it for this lab. Thank you.